As we've been speaking about the context of one molecule of glucose, and we're going to look at some other sources that can be metabolized to yield energy. So first of all, we can also metabolize glucose in the absence of oxygen. So we've been speaking about the electron transport chain, which we know requires oxygen, but understand that if we are in a state of deoxygenation temporarily, usually if we're doing high intensity exercise or if we're in the fight or flight mode, we can catabolize glucose in the absence of oxygen. This is called anaerobic glycolysis, and this relies on substrate level phosphorylation, which we talked about last time. So we basically back up the electron transport chain in a similar way that cyanide did. So if we don't have oxygen, once again, the electron transport chain grinds to a halt, and the Krebs cycle also grinds to a halt because there's not that final electron acceptor. So what we see is pyruvate building up, and as pyruvate is built up, it can be converted to lactate as a byproduct. And this is how we synthesize lactate when we catabolize glucose in the absence of oxygen. Okay. Remember, we don't have NADH because if we're not shuttling that NADH off to the electron transport chain because we're not using oxygen, then the NADH builds up as well, and that can be shuttled again through this lactate dehydrogenase pathway where we can make lactate as a byproduct. So this is a schematic representation of glycolysis. We know glycolysis yields 2 ATP as well as the reduced form of our carriers, which should go to the electron transport chain. But because there's no oxygen, the electron transport chain is not working. And so what we do is we shuttle pyruvate and the carriers that begin to build up. We shuttle them off to create lactate using the enzyme lactate dehydrogenase. And that is how we can yield some temporary ATP, okay? So we can continually cycle through this production of lactate and generate a small amount of ATP until we can fully oxygenate again and we can resume the electron transport chain. So it is a transient source of a small amount of ATP in the absence of oxygen, which kind of gets things going, but we know the much more efficient source of ATP is going to be oxidative phosphorylation in the mitochondria with the electron transport chain. Okay, so we're going to move on to speak about how the body can store energy using some of the other biomolecules that we mentioned. So we've been speaking about glucose, we're going to look at fats, we're going to look at proteins. Now we can store away energy for a later time. So let's say you just had a large, rich meal and you metabolize all that you need in terms of your energetic demands or your metabolic needs. You can store away whatever you don't need in the form of carbs, lipids, and proteins for use later on. So this is what we call the fed state. And in the fed state, once we have fueled our energetic demands, we store the rest of that away for when we are in the fasted state. Okay, so that's what we're going to speak about now. Now this first drawing, this first schematic shows the overall cycle of cellular respiration that we spoke about. So glycolysis to the linking step, to the Krebs cycle, to the electron transport chain. And we can see that other metabolites can enter this process as well. So proteins can be broken down into amino acids. What type of reaction is that? If we cleave a peptide down to its smaller amino acid components, is that a condensation or hydrolysis reaction? Are we using water or are we yielding water? This is the review, guys. It should be a review from what we spoke about last class, last week. Yep, Mackenzie. So condensation. Is it condensation? So we're breaking a peptide. We're putting in water. Hydrolysis, right? So hydrolysis. So we can hydrolyze our peptides down to amino acids. And then ammonia is going to be shuttled off to urea, which is going to be excreted through the kidneys. And we can enter this as either pyruvate, which can be metabolized, as acetyl-CoA, which can also be metabolized, or directly into the Krebs cycle. So this is how we can break down proteins and enter those substrates into one of three areas along the cellular respiration cascade. Likewise, we can shuttle to and from glucose and glycogen. So we know that glucose can be stored away as glycogen, usually in our liver and kidneys, sometimes in the muscle. And we can tap into that glycogen store. So back and forth, if we make glycogen, that's called glycogenesis. If we break it down to form glucose, that's glycogenolysis. And we could shuttle back and forth from glucose to glycogen, again, depending on our energetic needs, depending on our energetic demands at the time. And from glucose, we know that that can go directly to glycolysis, all the way down to oxidative phosphorylation. Likewise, triglycerides can be broken down. Now, there are two components to triglycerides. There's the glycerol backbone, and then there's the fatty acid tails, the three fatty acid tails. We can metabolize the glycerol backbone via glycolysis, and we can also metabolize those three fatty acid tails via the Krebs cycle. They can be entered via acetyl-CoA. They can be converted to acetyl-CoA, or they can be converted to coenzyme, which can enter the oxidative phosphorylation. So at various stages of cellular respiration, we can use our three biomolecules to sort of throw fuel into this process and generate ATP, okay? So let's start by looking at how glycogen is metabolized. So glycogen, usually stored in the liver and kidneys, can be broken down to form glucose, which can enter the bloodstream and make its way to other tissues and be used for energy. Now, in order for glucose to leave the cells of the liver, it must be in the form of glucose. Now, it's kind of stored temporarily as glucose 6-phosphate, but it cannot leave the cell in that way. So there are two mechanisms that can shuttle glucose to glucose 6-phosphate and back in order to provide a mechanism for glucose to leave the cell and be metabolized by other tissues. So only in the liver and kidney do we see this special enzyme that can turn glucose into glucose 6-phosphate and vice versa, so that it can be exported into the bloodstream and sent to other tissues. So basically we have a polysaccharide at the bottom. This is how glycogen is stored. And then we can break this down. It must be in the form of glucose to be exported from the cell to go into the bloodstream and make its way to other tissues to be metabolized. So we can synthesize new glucose as well. This is called gluconeogenesis. And we can make new glucose from glycerol, which is that glycerol backbone of fatty acids, from lactate, which we know builds up if we are metabolizing glucose anaerobically, or from amino acids that we break down protein in our diet from. So these are three of the other substrates that can generate glucose besides glucose itself. This occurs mainly in the liver, again, because this is where we have an enzyme present. And although we're making glucose from other substrates, this is not simply a reverse of glycolysis. There are two enzymes that are involved here, glucose 6-phosphatase as well as hexokinase, and they're the ones that are standing and sort of regulating the process of going from glucose 6-phosphate to glucose and back. So let's kind of illustrate that on this slide. So we're storing up glycogen, usually in our liver, sometimes muscle as well, and we can make glucose 6-phosphate from that glycogen, which can then do one of two things. It can be converted to glucose, it can leave the cell, and it can be metabolized in other tissues in our body. That is using the enzyme glucose 6-phosphatase, which is only present in the liver and kidneys. Okay? Or we can store this up by taking that glucose from the blood by the enzyme hexokinase, converting it back to glucose 6-phosphate, and then that can be stored as glycogen. So these two enzymes regulate whether or not glucose is being stored in glycogen or whether it's being metabolized, leaving the cell as glucose. Now from glucose 6-phosphate, we can either metabolize this via pyruvate, then the linking step, then the Krebs cycle, etc., or we can store it in our storage form of glycogen. But remember that if we convert glucose 6-phosphate to pyruvate, it can only be used in the liver cells. themselves. If we need it to get out into the blood and to other tissues, it must be in the form of glucose via this enzyme glucose 6-phosphate.